recognize in evolution, and then move on to where is he? Oh, he escaped. He has changed his place. Where is he? You know? Oh, no. Ah, he is here only. Sorry. You were sitting there yesterday. So I assumed because he was not there, he would be somewhere in between. Adaptive radiation we discussed. Biological evolution. Evolution by natural selection in the true sense would have started when cellular forms of life with differences in metabolic capability originated on Earth. What? Adaptive radiation we did not. What is adaptive radiation? Okay. What is adaptive radiation? See, adaptive radiation, should it be, we'll go again into what it is, but should it be divergent or convergent? Adaptive radiation is noticed in divergent evolution, option A, option B convergent evolution, option C both, option D neither. Divergent. Okay. Why? Because it is radiation. Radiation is moving from one place. Okay, adaptation is a cause of variation in convergent evolution, divergent evolution, both, neither. Adaptation is a contributing factor for evolution in which of these two? Convergent evolution or divergent evolution? Both, neither of them. Both. Why would the organisms with similar structures develop different functions? To adapt to the condition where they are existing. Why would organisms, that is divergent evolution, the development of homologous organs. Why would organisms with different structures develop similar functions in a similar kind of environment? That is also because of the forces which drive them to adapt. All right, anyways. During his journey, Darwin went to Galapagos Islands. There he observed an amazing diversity of creatures. Of particular interest, small black birds, later called Darwin's finches, amazed him. He realized that there were many varieties of finches in the same island. All the varieties, he conjectured, evolved on the island itself. From the original seed-eating features, many other forms were with altered beaks arose, enabling them to become insectivorous and vegetarian finches. This process of evolution of different species in a given geographical area, starting from a point and literally radiating to other areas of geography or habitats, is called adaptive radiation. Darwin's finches represent one of the best examples of this phenomenon. Another example is Australian marsupials. A number of marsupials, each different from the other, evolved from an ancestral stock, but all within the Australian island continent. When more than one adaptive radiation appeared to have occurred in an isolated geographical area, representing different habitats, one can call this convergent evolution. Placental mammals in Australia also exhibit adaptive radiation in evolving into varieties of such placental mammals, each of which appear to be similar to a corresponding marsupial. Example, placental wolf and Tasmanian wolf marsupial. See, now there is a point to highlight here. All the finches that Darwin recognized or identified were on the Galapagos Island. All of them belong to the same island. But they have developed varieties. How? Divergent evolution divergent evolution in a continuous or in a geography which is in continuity. How do you explain that? It is on the same island, no? If they were all seed-eating ones, if they were all seed-eating birds originally, what forced them to develop insectivorous habit favoring beak?
Maybe seeds were limited.
Seeds were limited, okay. So that is one force for survival. There is a drive. Competition is driving them into try different things. That is one thing. Can it also not be the other way? Isn't this the same example which can be used to justify the mutation theorists also? De Vries supporters also? Mutations had happened in these birds which made some of them develop a little more pointed beaks, which gave them the advantage in eating insects as well from the tree trunks. That is also possible, no? So Darwin there is a drive, there is a force to adapt, and that force will drive adaptation, which brings about phenotypic modification, justified by genotypic alteration. The other way is also true. The other way also can be logically explained. There are changes happening continuously at the genotype level, at the gene level, and those changes, if it is corresponding to a particular phenotypic variation, would give rise to that phenotypic variation. Incidentally, that phenotypic variation could be advantageous in a particular situation. And those with the phenotypic variation have more likelihood of survival in that particular situation, making the population of those kind of organisms more, because they have a selective advantage. You see the continuity of thought. You started with evolution, you started with explanation by using the mutation theory. Then their prominence in a particular geography can be explained by using Darwin's theory of natural selection. You get that? Again, remember the elephant story. Everybody is talking about the same thing from different perspectives. Darwin is talking about the survivability of organisms based on the advantage they have because of some adaptation. Darwin says it is the forces which threaten their survival that drive them to develop these characters. De Vries accept that the advantage is allowing them to survive. But the cause for those changes is not this, which is a very slow process spanning several generations and passing on the graded incremental change in genes which will eventually accumulate into an advantage is not explainable as per De Vries theory. And he says the change is sudden. The change is sudden. Adaptation and dominance will happen over time. You get that? So convergent evolution, divergent evolution, adaptive radiation, these are all just explanations given for us to easily categorize concepts. Otherwise, everything is in continuity. All forces are in play. Okay. And being in a geographical area, in the same geographical area, convergent evolution. The example is given of Tasmanian wolf and Tasmanian wolf, which is a marsupial, and the placental wolf. Both of them developing similar characteristics by virtue of them surviving in a particular geographical area. And marsupial mammals also developing placental mammal-like characteristics when they are supposed to survive in an area which is different from this place where they originate from. All right. Biological evolution. Evolution by natural selection in a true sense.
What are marsupials?
Marsupials are the ones which have an intermediate stage of development, pouched mammals. Okay. Kangaroo. They have a pouch in which a partially developed embryo is nurtured before it completely becomes before its free existence. Okay. So it's like internship for the real world. What happens in the womb or the uterus is the course, 4.5 years of MBBS. What happens in the pouch is 1 year of internship before you get out into the world to face the real world independently. For human beings, internship goes on for 18, 20, 25, 30, sometimes lifetime, where parents take care of their children, continue to take care of their children forever and ever. All right. Ideally by 18, 20, at least by the time you finish your graduation, internship should complete, social internship. But that continues for many people for lifetime, and parents, some of the parents pride themselves about it. Don't worry, I will take care of you till I am alive. Okay. Okay. Biological evolution. Evolution by natural selection in a true sense would have started when cellular forms of life with differences in metabolic capability originated on Earth. What does that mean? See, natural selection is not a conscious process. Natural selection is an observed phenomenon. It is not like now natural selection will come into picture and this will happen. You observe something happening and you can say this has happened because of natural selection. Selection here is kind of a misnomer in my opinion. Nature is not selecting. It is just that those with an advantage are able to survive. Those without the advantage are not able to survive. You can at best say, you can as well say natural elimination, no? If you can say natural selection, nature is selecting the stronger ones. As a consequence of, as a corollary to that, you can say natural elimination, nature is eliminating the weaker ones. Whereas in reality, nature is neither doing selection or elimination. Nature has created conditions. Nature has not even created conditions. There are natural conditions. Against those conditions and in adaptation to those conditions, organisms have to survive. Those which have the adaptation to survive will survive. Those which do not have the capability to survive will vanish. They will die. Birth, survival, existence and death are all natural processes that happen. Those that adapt will not survive forever. Those that perish would, it's not that they have not survived at all. They perished as soon as they are born. So all of these are happening. All of these are happening. It is just that with advantage gives better survivability, disadvantage reduces the survivability chances. Those who can survive better have more probability of accessing resources or accumulating resources. Those with better access to resources have the ability or have the higher probability of reproduction. So take this over a period of time. Those with adaptation have better resource availability or access, which reproduce more. So gradually the number of individuals of that population which has the adaptation will increase, and the size of the population of that with a disadvantage will decrease. When it reaches a critical mass, when it crosses a certain stage, this one can completely get extinct. And everybody in the population will be the one with this adaptation. Now there is no advantage. When everybody in the population have a particular adaptation, the adaptation is not an advantage anymore. For some other condition, some other challenge, some other adaptation comes in a set of people or set of individuals in that population, that is an advantage again. Then again the natural forces will come into picture. You understand? So this started happening even from the early stages of cellular development, where, let's say, currently, enzymes are the catalysts of metabolic reactions. Enzymes are the catalysts of metabolic reactions. We also see RNA being catalyst. Where is RNA a catalyst? Where do you find RNA as the catalyst? Ribozyme. Where? In translation process. In translation process, the peptidyl transferase enzyme. Do you remember? Peptidyl transferase enzyme will transfer the amino acid from the tRNA on site A to the tRNA that comes to site, that from the one which moves from site P to E to the one which moves to site P. Peptidyl transferase is contributing to joining the amino acids and forming the peptide bonds attached to the in the amino acids attached to the tRNA that is in site P. Correct? Peptidyl transferase is an RNA enzyme, ribozyme. That means right now also we have the evidence of RNA also being a catalyst. RNA also being a catalyst. But why do we have almost all the enzymes except a few exceptions being proteins? Natural selection. The ability of proteins to catalyze chemical reactions was much better than the ability of other biological molecules to catalyze reactions. So eventually the reactions that were catalyzed by proteins became more prominent, predominant reactions, and those which were catalyzed by others started vanishing. Natural selection. Why ATP the energy molecule? Why not something else? Natural selection. That is the meaning of the first statement there. The essence of Darwinian theory about evolution is natural selection. The rate of appearance of new forms is linked to the life cycle or the lifespan. How fast a new organism will evolve is dependent on how fast the new organism reproduces. And how fast a particular organism reproduces is dependent on how long is its lifespan. Something which lives very long, its reproductive cycle is long. So the change of generations is stretched. But those which will reproduce faster, the lifespan is shorter, and the change of generations happen quick. Evolution is change over generations. So if you can get 100 generations in 2 days, as compared to one generation in 50 years, over 50 years, the evolution of the one which is multiplying so fast will be much more than the evolution of the one which reproduce, where generational change happens every few years. Right? So the rate of appearance of new forms, that is adapted, that is new species or new varieties, is linked to the life cycle or the lifespan. Microbes that divide fast have the ability to multiply and become millions of individuals within hours. Colony of bacteria growing on a given medium has built-in variation in terms of ability to utilize a feed component. A change in the medium composition will bring out only that part of the population that can survive under the new conditions. In due course of time, this variant population outgrows the others and appear as a new species. This would happen within days. That's all, whatever we discussed now. They are capable of producing new generations very fast. So within a few days, you have the possibility of creating new species. The implication of this is, the understanding of this should tell us that our attempts to eliminate microbes by the use of antibiotics or other chemicals is only contributing to better evolutionary ability in them. We are contributing to their strength. We are making them stronger. Correct? And our body, our immune system is capable of fighting the diseases caused by organisms to which we have been exposed over the entire evolutionary history of humans. For millions of years, whatever we have been exposed, the immunity has developed to that, and a collective immunity, innate immunity, what we speak about. Innate immunity is it's like antivirus in your computer. It keeps updating, no? So the new generation is getting the updated copy or updated software of all the things that we have fought in the past. But earlier, evolution of the attackers, if we consider that, disease causing microorganisms, if we consider as our attackers, the evolution of attackers used to happen by conflict with natural forces. The bacteria should infect human beings. There is a fight between the human immune system and the bacteria. In that, so many bacteria will die. Some of them will develop some adaptation. They have to pass on to another host or to another medium to be able to thrive. And then they have to get into another person to further grow and multiply. This was taking a lot of time. Now what we are doing, with the abuse of antibiotics, with the abuse of chemicals that are designed to kill this bacteria, we are constantly training them. It is training them. Vaccination is training our immune system to develop ability to fight a particular organism. Antibiotics are like vaccinating bacteria. In one way, it is killing them. But some of them are getting vaccinated. That means they will develop resistance to that. Okay. So, how do I say this? There is a character that comes in our history, a demon. His name is Raktabijasura. Raktabijasura. He is a demon. He has a special ability. The ability is, if he is cut and his blood spills, for every drop of blood, he clones. So if you spill 100 drops of blood from his body, you will have 100 Raktabijasuras. You cut each one of them, for every drop, each one of them will give rise to one. So you cannot kill him by spilling his blood. If you want to kill him, you cannot spill his blood. That is the challenge. Similar to antibiotic resistance. Don't use antibiotics to kill bacteria. All right. For the same thing to happen in a fish or fowl would take millions of years, as lifespans of these animals are in years. Here we say that fitness of B is better than that of A under the new conditions. Nature selects for fitness. One must remember that the so-called fitness is based on characteristics which are inherited. Okay. Hence, there must be a genetic basis for getting selected and to evolve. Genetic basis for getting selected and to evolve. Another way of saying the same thing is that some organisms are better adapted to survive in an otherwise hostile environment. Adaptive ability is inherited. It has a genetic basis. Fitness is the end result of the ability to adapt and get selected by nature. Same thing what we discussed. See, the adaptation, what you have, should have a genetic basis. And the genetic basis is based on what you have inherited. It cannot be based on what you have developed in this generation, in your own lifespan. That is also phenotypical adaptation. It is not inherited adaptation. Okay? If you are supposed to climb mountains, somebody who is born with a strong skeletal system and a strong muscular system by inheritance would have an advantage in climbing mountains. Somebody who is born with not so strong skeletal and muscular system can also develop the ability to climb mountains by training. He is enhancing the ability by practice and training. That is there. But it is not natural selection, it is by effort. Alright. So the next generation of these two people, the one who was born with a stronger skeletal and muscular system, their next generation also will be born with a strong muscular and skeletal system. But the one who trained himself to become strong, his next generation will again be born with what his genetics allows him to be born with, allows them to be born with. They will also again have to train. All right. Branching descent and natural selection are the two key concepts of Darwinian theory of evolution. What is branching descent? Branching descent, adaptive radiation. Originate from a common area, branch out into different areas, adapt to those areas, and the change happens because of these adaptations. Even before Darwin, a French naturalist Lamarck had said that evolution of life forms had occurred but driven by use and disuse of organs. He gave the examples of giraffes who, in an attempt to forage leaves on tall trees, had to adapt by elongation of their necks. As they passed on this acquired character of elongated neck to succeeding generations, giraffes slowly over the years came to acquire long necks. Nobody believes this conjecture anymore. See, for any somatic change to be transmitted to the next generation, that change has to be represented in the genes of the reproductive cells. All right, that is also validated. There are experiments to prove that this is also possible. All right, those potato experiments, not given in this, but there are experiments to prove that this is also possible. Somatic changes being passed on to the next generation, but that is subject to those changes being reflected in the gametes also. Now let us say continuous exposure to sunlight. Continuous exposure to sunlight will change a particular gene in the skin cells, will change a particular gene in the skin cells, which makes the person more susceptible to developing skin cancer. Let us assume that. Isn't that gene present in the gametes also? Isn't that gene present in the gametes also? The entire genetic makeup is present in every cell. So what if the stem cells in the gonads also develop the same mutation? If the stem cells in the gonads also develop the same mutation because of exposure to sunlight, which makes a person susceptible for skin cancer, that can be passed on to the next generation. So there is a possibility that nobody in the ancestry had skin cancer, but somebody in the subsequent generations might develop skin cancer. How do you explain that? Nobody in the past had diabetes at all in the family, but somebody might start developing diabetes. Diabetes has a strong genetic predisposition. So does that mean this person developed diabetes because of his bad habits? He brought it upon himself? No. Susceptibility. There could have been modification. All right. Somebody who has never been a smoker. Have you seen Munna Bhai MBBS movie? Munna Bhai MBBS? No? If you have not watched, you should watch it on one of the Sundays. There are two movies, Munna Bhai MBBS and Lage Raho Munna Bhai. Nice comedy movies. If you have not watched Munna Bhai MBBS, then the example will not relate. There is one character that comes in the movie, Zahir. Zahir is the friend of Munna Bhai. If I use this example now, those who have not watched the movie will have a problem. Anyways, it's okay. I watch suspense movies with climax being known also. Murder mysteries, I know who is the murderer and I'll still watch the movie. So I don't mind spoiling suspense for all of you. Zahir develops lung cancer. Zahir develops lung cancer. He doesn't smoke, he doesn't drink, he has all the good habits in the world, but he develops lung cancer. There are examples that in the same village, there is a 90-year-old man who is a chain smoker who smokes about 20, 30, 40, 50 BDs every day. He keeps smoking. He is like a chimney, continuously smoking out smoke. He lives to dry out like a leaf and die. But there might be a 40-year-old man or a 35-year-old, 50-year-old man who has never smoked, who has no bad habits, dying because of lung cancer. And that fellow says, see, it is not BD. Okay. It is not BD that causes lung cancer. He will validate his own claim. Why? Genetics has two components, nature and nurture. The genes will give you the susceptibility. The exposure to factors that can trigger the manifestation of the gene gives you triggers. Let us say X and Y are the two factors which will determine lung cancer. X is susceptibility. If somebody is susceptible by 10 times more than somebody else, so susceptibility of person A is 1, susceptibility of person B is 10. The first person has to smoke 100 cigarettes a day to develop the susceptibility of 100. To develop the combined effect of 100. Let's say 100 is the limit to reach to develop lung cancer. The first fellow should develop 100 cigarettes a day to get lung cancer. The second one has susceptibility of 10. Even if he smokes 10 cigarettes a day, he will develop lung cancer. Suppose somebody has a susceptibility of 50. Smoking equivalent to 2 cigarettes a day might cause lung cancer in them. And smoking equivalent to 2 cigarettes a day would happen by passive smoking. Alright, you don't have to smoke, passive smoking. So somebody who has never smoked develops lung cancer. All possibilities, all possibilities. So it's not just one factor, two factors. It is multiple factors. When we study one factor to understand it in simple ways. So Lamarck use and disuse theory. Is evolution a process or the result of a process? That is what we discussed. The world we see, inanimate and animate, is only the success stories of evolution. When we describe the story of this world, we describe evolution as a process. On the other hand, when we describe the story of life on Earth, we tell natural resources are limited, populations are stable in size, except for seasonal fluctuations. Members of a population vary in characteristics, even though they look superficially similar. Most of the variations are inherited, etc. The fact that theoretically population size will grow exponentially if everybody reproduced maximally, this fact can be seen in a growing bacterial population. And the fact that population sizes is in reality are limited means that there had been competition for resources. Only some survived and grew at the cost of others that could not flourish. The novelty and brilliant insight of Darwin was this. He asserted that variations which are heritable and which make resource utilization better for few, adapted to habitat better, will enable only those to reproduce and leave more progeny. That is what we discussed. The variations give advantage, advantage gives them better resource utilization gives them better reproduction ability. They are the ones who are likely to produce more progeny. So their generations will flourish. Those with the disadvantage, their generations will decline. Hence, for a period of time over many generations, survivors will leave more progeny and there would be a change in population characteristic and hence new forms appear to arise. This is how demography changes. Those who are constantly becoming powerful populations, such populations will become more and more over a period of time and they will become the dominant population. There is always a threshold. Or a critical mass for any demographic change. Once the critical mass is crossed, the change will be faster. Alright? So if you look at the history of life on this planet, communities like America. Today America is a country with a certain set of population, different kinds of population. But there was a period of time when the entire landscape of America was occupied by, there were Native Americans living there. Today if you go to America, you have to go to a particular place as a tourist to see how Native Americans looked. Very few of them are surviving. Whatever the reason, you go to any place, you look at the history of 1,000 years of that place and see how the nature of population has changed in that place over 1,000 years. 1,000 years is a very small time for life. 1,000 years is a very small time for the history of life. But even if you study history of 1,000 years of any given place, take any country for that matter, go back into a 1,000-year history. What was the kind of population that survived there? What were their practices? What were their habits? What were their belief systems? With every 100 years, things are changing. And what appears to be the normal today might not have been 1,000 years ago. So cyclical, it keeps changing. Natural selection process. Humans will complicate it because they will bring in their own influences into demography. What is demography? Demography is the composition of populations in a particular place. That is demography. Okay, when we talk about the demography of professions in Bangalore, you will talk about the different kind of professions that people are practicing in that particular geographical area. If you talk about the demography of population based on faith or belief systems, then you have different belief systems represented by different set of people. That is how you describe demography. All right. Demography is a very, very important term. Mechanisms of evolution. What is the origin of this variation and how does speciation occur? Even though Mendel had talked of inheritable factors influencing phenotype, Darwin either ignored these observations or kept silence. In the first decade of 20th century, Hugo de Vries, based on his work on evening primrose, brought forth the idea of mutations, large difference arising suddenly in a population. He believed that if a mutation which causes evolution, it is mutation which causes evolution and not the minor variations that Darwin talked about. That is the major point of conflict between de Vries and Darwin. They did not fight, but de Vries disagreed with Darwin on the point that evolution is not happening because of minor variations accumulated over a period of time. It happens suddenly, only because of mutations. Mutations are random and directionless, while Darwinian variations are small and directional. This is important. This is a point that will be highlighted in questions. Mutations are random and directionless. Darwinian variations, what Darwin speaks about, are directional. It is like, see, which of these two can be considered to be driven by purpose? Darwin's theory is, or De Vries theory is based on purpose-driven evolution, if you have to interpret it that way. Blanket you need? Getting too cold, not getting enough sleep. Huh? Drink water. Don't sleep. He has covered completely, wrapped up in muffler. Only thing pending is blanket and a convertible bed. Huh? Drink water. Which one is purpose driven? Darwin's theory or De Vries theory? Purpose driven means, if I have to say, evolution happens for a purpose. Darwin's theory. Because Darwin speaks about evolution moving towards providing an organism an advantage to survive. Purposeless is De Vries. De Vries says it happens. It happens in all directions. There can be beneficial mutations, there can be harmful mutations, there can be lethal mutations. Mutation that will give advantage, mutation that will give disadvantage, mutation that can kill also. And mutation that doesn't do anything. So in molecular theory also we discussed that, no? Point mutation, frame shift mutation. Point mutation and frame shift mutation. In point mutation or frame shift mutation, we also have same sense mutation, missense. In point mutation, you have same sense, missense, and nonsense mutation. So this is happening at the molecular level. We are talking about it in the molecular level. At the phenotypic level, if the mutation causes some visible change phenotypically or physiologically, functionally, then that will either give advantage or disadvantage or nothing, just like that. So De Vries is purposeless. What do you think? Whom do you want to support if it is a vote between De Vries and Darwin? How many of you will vote for De Vries as your thought leader? Choose one. How many of you vote for De Vries? Raise your hand. Oh. How many of you vote for Darwin? And true to Indian democracy, most of the people don't vote. Okay. Out of 48 eligible voters, we had 5 people voting for De Vries and 3 people voting for Darwin. 40 people did not vote. 15% voter turnout. And that too when all the voters are in this room and the effort that was required was just to raise hand. Imagine the effort required to get out of your house on a holiday, go to the polling booth, stand in the queue. Oh my God. That is near death experience, no? Okay. Participation. Okay. I think Germany has participatory democracy. What is participatory democracy? We have electoral democracy. If I am not wrong, Germany has participatory democracy. Democracy itself is participation of people. Then what is participatory democracy? Huh? Innocence is bliss. I am making it sugar-coated. The actual statement is ignorance is bliss. Okay. What is the difference between innocence and ignorance? Both are a state of not knowing. A state of not knowing can be called innocence or ignorance. Innocence is a state of not knowing in a person who does not have the ability to know. A child is innocent. An adult cannot be called innocent. An adult is ignorant. All right. A child does not know. A child is not in a position to know things because the child has limited exposure, limited tools and resources. The child only has curiosity, which will be successfully stifled by adults in the family around. The child start asking questions. How many questions do you ask? Shut up. Don't ask questions. Okay. Electoral democracy is people vote. They choose their representative, and the representatives collectively will make decisions for all the people together. For every decision, people are not participating. Not possible to have people participating for every decision. Then every decision will become like an election. In smaller democracies, people will participate. They will vote on policies. They will first elect their representatives, and the representatives will put out a list of options in front of the people for every policy, and the people will vote for their respective choices, and that will also be considered while making decisions. That is participatory democracy. Not relevant to the discussion that we are having, though it is very, very relevant. Okay. Evolution for Darwin was gradual, while De Vries believed mutation caused speciation and hence called it saltation, single step large mutation. Studies in population genetics later brought out some clarity. Hardy-Weinberg principle. We have discussed it in detail, but just we'll go through it. In a given population, one can find out the frequency of occurrence of alleles of a gene or a locus. This frequency is supposed to remain fixed and even remain the same through generations. Hardy-Weinberg principle stated it using algebraic equations. This principle says that allele frequencies in a population are stable and is constant from generation to generation. The gene pool is the total genes and their alleles in a population. Okay, gene pool is not genotype. Gene pool includes all the alleles for a particular gene in a population. So the gene pool of human beings will have what alleles for blood group? How many alleles are there for blood group in the gene pool of human beings? How many alleles are there for blood groups in the gene pool of human beings? 3. Which are? IA, IB and small i. 3 alleles are there in the gene pool of human beings for blood group. How many alleles are there for blood groups in the genome of human beings? 2. Genome is of individuals. All right. Gene pool is for the entire population. Gene pool represents all the variations of alleles, all the variety of alleles that you have for a particular gene. But genome represents the number of alleles that will be there for a particular gene in the population. Okay, right. The gene pool remains a constant. This is called genetic equilibrium. What is genetic drift? Genetic equilibrium is the gene pool remains constant. There is no change in the gene pool, either in terms of the number of alleles that are present in the gene pool or in terms of their relative frequencies, their relative composition. Okay, we'll come there. Sum total of all the allelic frequencies is 1. Individual frequencies, for example, can be named P, Q, etc. In a diploid, P and Q represent the frequency of dominant allele and recessive allele. Capital A is dominant allele, small a is recessive allele. The frequency of homozygous dominant individuals in a population is, what is it? P square. This is simply stated in another way. That is the probability that an allele A, capital A, with the frequency of P appears on both the chromosomes of a diploid individual is simply the product of the probabilities, that is P square. Similarly, of small a is Q square, of capital A small a is 2pq. Hence, P square plus 2pq plus Q square is equal to 1. This is a binomial expansion of P plus Q whole square. When frequency measured differs from expected values, the difference indicates the extent of evolutionary changes. See, how do we know that the evolution has happened? Let's say we know the gene frequency. We know the gene frequency for a particular population for generation number 1. We measure the gene frequency for a particular population for generation number, let's say 100. And the gene frequency has changed. If the gene frequency has changed, that means there are some evolutionary change that has happened. This is all theoretical. We cannot do it for human beings, human populations. You can only do it experimentally for controlled populations. To prove these theories, you will do it for controlled populations. You will do it in controlled populations. In a natural environment, you will not be able to do this because the principle that drives Hardy-Weinberg equilibrium is geographical isolation, which is not seen in nature. Absolute geographical isolation to have genetic equilibrium is not found in nature, we can say to the largest extent, unless there is an unexplored island populations, geographically isolated populations. If there is geographical isolation for a population, that means the population is on an island. Nobody has gone to that island, nobody has come out of that island. Such populations you will find genetic equilibrium. Otherwise, where there is migration, where there is influx and efflux of population, you will not find genetic equilibrium. It's like the sum total of all energy is the same, is always constant. So this is also like that. 5 factors are known to affect Hardy-Weinberg equilibrium. Important. 5 factors are known to affect Hardy-Weinberg equilibrium. These are genetic migration or gene flow, genetic drift, mutation, genetic recombination, and natural selection. Okay. Component you have to remember. These 5 factors you have to remember. Gene flow is gene migration, that is people moving away from one place to another place. Genetic drift is change of genetic expression. Genetic recombination, mutation, genetic recombination, all of that we know, natural selection. When migration of a selected population to another place and population occurs, gene frequencies change in the original as well as in the new population. New genes or alleles are added to the new population and these are lost from the old population. There would be a gene flow if this gene migration happens multiple times. If the same change occurs by chance, it is called genetic drift. Alright? Gene flow is the same as genetic drift. Gene flow is by chance or by design. That is gene flow. But gene flow happening by chance is genetic drift. Alright? Sometimes the change in allele frequency is so different in the new sample of population that they become a different species. The original drifted population becomes founders and the effect is called founder effect. Founder effect. What is bottleneck effect? There is one more effect called bottleneck effect. What is bottleneck effect? Founder effect, you might have come across questions on bottleneck effect. Not yet so probably. Founder effect is a set of individuals from a population migrate to a new population, hitherto not having this set of genes. So far this new place does not have these set of genes in the population, in the geography. These are the people who will go with this new set of genes and they are the ones who will be the founders of the new genetic pool. So that is founder effect. Bottleneck effect. What is a bottleneck? When we talk about traffic, what do you refer to as bottleneck in traffic? A bottleneck in traffic. Something wider gets narrow. Alright? Like this. This is a flask neck. Okay. Bottleneck effect. Let us say there is a large number of genes present in the gene pool. But because of whatever factor, a set of genes are eliminated from the population and the expression happens only of a few variations of the genes in the gene pool. Can happen because of catastrophes leading to killing of populations, disasters, genocides, whatever. You eliminate a set of individuals from the population by natural causes or whatever cause, the number of genes that can express or the diversity of genes in the gene pool will become lesser, causing the expression of the genes to a limited extent compared to what it was originally. Bottleneck effect. Limiting the expression of the variations of the gene from the gene pool. What limits it? Whatever it can be. Alright? Have you seen Game of Thrones? Anybody? There is that one group of soldiers, mercenaries. Not mercenaries. Who are they? The most loyal fellows who will follow the master. The dragon queen takes them from the masters. The unsullied ones. The unsullied population. Bottleneck effect. Why? The unsullied cannot reproduce. All of them have been neutered. So they are present in the population, but the gene variations that they represent cannot pass on. The expression is restricted by those unsullied soldiers being neutered, castrated. Alright. So that is bottleneck effect. There is founder effect also. There is founder effect also in Game of Thrones. So, anyways, let us not go into that. Most of you don't know what happens there. So I have also forgotten the entire plot long back, 2012, 13, 14, somewhere around that time. So you watched it when you were in primary school. Huh? You're not supposed to watch it in school. Okay. Right. Microbial experiments show that pre-existing advantageous mutations when selected will result in observation of new phenotypes. Over few generations, this would result in speciation. Natural selection is a process in which heritable variations enable better survival, are enabled to reproduce and leave greater number of progeny. Same thing repeated again and again and again and again in different ways. See, you tell me, what are all the factors which can lead to the formation of new species? Tell me, you put down the factors which can lead to the formation of new species. Gene flow, gene migration, genetic drift, natural selection, mutations. Recombination will lead to variation. And isolation is necessary for speciation. Yes or no? There should be no recombination. Recombination is against speciation. If recombination continues to happen, there will be variations produced, not speciation. Isolation is necessary for speciation. New species to be formed. Alright. Isolation, natural selection, genetic drift, all these can contribute to species. Mutation is a significant factor. So it's like you keep a set of people isolated with no interaction with the rest of the world for hundreds of years. They will appear like a different species. That is what you see with native populations, tribal populations which do not have connection with the rest of the world. Who is better, who is not better, what is more advanced, what is less advanced, all of those are different things. There is a movie where the Britishers go, not the Britishers, I think American colonizers go to a particular place. They talk to the leader of that tribe. You've seen that? Movie or series?
It's about the Hawaiian Islands.
So they tell him, you give up your land and you will be in the protection of the American government. He says, we don't want your protection. We will give you something better. Better in comparison to what? Better in comparison to what you already have. We are happy with what we have. We don't want something else. So how do you colonize or how do you enslave a content population? If you are content, if you are happy, you cannot be enslaved. If you can be enticed, you can be enslaved. Enticement is a tool to enslavement. You fall for it, you can be enslaved. Got it? Okay. A brief account of, okay, this is. A critical analysis makes us believe that variation due to mutation or variation due to recombination during gametogenesis or due to gene flow or genetic drift results in changed frequency of genes and alleles in future generation. Coupled to enhance reproductive success, natural selection makes it look like different population. Natural selection can lead to stabilization in which more individuals acquire mean character value. Stabilization of the population. There can be stabilization on either way sides. See, you see the diagram given on the other page, figure 6.8. Stabilizing selection, directional selection and disruptive selection. Those are the three types of selection that is given. Okay. The first one is a particular phenotype. Phenotype favored by natural selection is the mean. So what is the maximum number of individuals you find in a particular population for a particular trait? That is the trait that has been selected naturally. So that is the one which has the advantage. Now the selection can be directional. If you see A, it is stabilizing selection. That means the peak gets higher and narrower. A narrow band of traits are selected. Like this. We'll go this way. This is the population that we find. All right. Now you consider this is the band of population that is selected. This is the extremes. These are the extremes. Stabilizing selection is a particular character is more advantageous. So the number of people with a particular character becomes more and the rest of the population gets wiped out, or the characteristic expressed by the character expressed by the other population or the other character expressed by the population is vanished. So this was the band earlier. The number of people were more in a wider frame of characters. Now this particular narrow band of character is selected. But the number of people will remain the same, no? Or more or less the same. So this peak increases. This is stabilizing selection. Character which is the most advantageous will be expressed by most of the people in the population and the extremes become less. So most of the people will stabilize with that particular character. This is stabilizing selection. Another selection is directional selection. Suppose, now this one, the black band is what you see. Let us assume this particular character, the character expressed by this population is the more advantageous one. So eventually over a period of time what will happen? The population will shift towards that direction. So the character that was expressed by a smaller group of people earlier, because that was the one which gave advantage, will shift to the direction, will shift in that direction. Alright. So let us say this is of height. This extreme represents dwarf people. This extreme represents very tall people. This extreme represents, this mean represents the people with moderate height. Let us say this is for tall people. It becomes advantageous for tall people to survive better. So over a period of time, natural forces or the natural selection forces will make it such that the people who are very tall, above 6 feet, become more in the population. Then even the ones with shorter height become lesser and lesser. Mean height become lesser and lesser. This is directional selection, where one of the extremes becomes more advantageous. The mean shifts towards that extreme. Disruptive selection, if you see this one, see, disruptive selection is, the middle part is not useful. It is good to survive if you are less than 4 feet tall or more than 6 feet tall. People with 4 to 6 feet height don't have that advantage. Whatever the condition, you can imagine the conditions. Either you have to go in some tunnel and get something, or you have to reach out to something. If you are of the moderate height, you can neither go into the tunnel nor reach out. So you will not survive. That is disruptional selection, where the extremes the extremes are selected and the middle part is not selected. There is disadvantage here. Being average, being in the middle is not advantageous. Either this extreme or that extreme. So in disruptional selection, you will have two peaks. You will have two peaks in the extremes. One in each of the extremes. You understand that? Consider this as height, basically. Now the directional selection need not be only on this side. It can be on this side also. It can be on this side. Gulliver's Travels. You know that? Gulliver is an average height man. He is a normal man in his place. He is the average man in his place. He goes on a voyage and he accidentally gets into a place which is of Lilliputs. Lilliputs are dwarfs, very dwarf people. For them, this fellow appears like a huge monster, giant. They can climb on him and all. So probably in the Lilliput land, being that short is advantageous. So if this fellow Gulliver continued to stay there, his 100 generations continue to stay there, over a period of time, his future generations will start becoming shorter and shorter if they have to survive. Otherwise, they will not survive if Darwin's principles work. So the selection can be directional in this side also. Okay, let us say food has to be got by crawling through tunnels. In such places, shorter people will be dominant. Food has to be obtained by reaching out to higher plants, higher trees, taller trees. In that places, this will be selected. If both options are available, then it can be this one. We are just talking about height. For every character, natural selection forces work like this. So you have to remember three processes of natural selection. One is, what is the first one? Where it becomes narrow. Stabilizing selection, where the mean is stabilized and most of the population will arrive at that mean expression. Directional selection, where either of the extremes is favored. Disruptive selection, where both the extremes are favored and the mean is eliminated. Understand? That is what the diagram gives you. Okay. A brief account of evolution. Now we are going into specific evolution of organisms. About 2000 million years ago, the first cellular forms of life appeared on Earth. The mechanism of how non-cellular aggregates of giant macromolecules could evolve into cells with membranous envelope is not known. Some of these cells had the ability to release oxygen. The reaction could have been similar to the light reaction in photosynthesis, where water is split with the help of solar energy captured and channelized by appropriate light harvesting pigments. Mix up everything, current knowledge, and build a theory. All right. Slowly, single-celled organisms became multicellular life forms. By the time of 500 million years ago, remember these timelines. Single-celled organisms came around 2000 million years ago. About 500 million years ago, invertebrates were formed. Jawless fish probably evolved around 350 million years ago. Don't bother about anything else. Whatever is mentioned here, 2000 million years ago, first life form. 500 million years ago, invertebrates. 350 million years ago, jawless fish. Seaweeds and few plants existed probably around 320 million years ago. We are told that the first organisms that invaded land were plants. So this is also important. The first life form to evolve on land was plants, invertebrates, vertebrates, amphibians. These all can be options. Here it is given, it is plants. Alright. These things you have to just pick up. If you start paying attention to the book, you will realize what can be questioned. Exceptions, something unique, something that can confuse, something which can go against popular logic. Alright. They were widespread on land when animals invaded land. Fish with stout and strong fins could move on land and go back to water. This was about 350 million years ago. In 1938, a fish caught in South Africa happened to be a coelacanth, which was thought to be extinct. These animals, called lobe fins, evolved into the first amphibians that lived on both land and water. The ancestors for amphibians was lobe fins. That can be the question, no? The evolutionary ancestors for amphibians were lobe fins. It can be fishes, jawless fishes, cartilaginous fishes, lobe fins. Some rest of the things can be cooked up. There are no specimens of these left with us. However, these were ancestors of modern day frogs and salamanders. The amphibians evolved into reptiles. They lay thick-shelled eggs which do not dry up in sun, unlike those of amphibians. Again, we only see their modern day descendants, the turtles, tortoises and crocodiles. In the next 200 million years or so, reptiles of different shapes and sizes dominated on Earth. Giant ferns or pteridophytes were present, but they all fell to form coal deposits slowly. Some of these land reptiles went back into water to evolve into fish-like reptiles probably 200 million years ago. Ichthyosaurus. The land reptiles were of course the dinosaurs. The biggest of them, Tyrannosaurus rex, was about 20 feet in height and had huge, fearsome dagger-like teeth. How did they appear and what was their behavior and all that? You can see in Jurassic Park. Okay. T-Rex. What others have come in Jurassic Park? One is T-Rex. The other one, the smaller ones were? Velociraptors. Those are the ones which are trainable, no? They train the Velociraptors to go and attack the T-Rex. Finally, I think they have brought in some emotions also to T-Rex. Yes. About 65 million years ago, the dinosaurs suddenly disappeared from the earth. Now you have to correlate. 65 million years ago, what was the time? What was the time between the Cretaceous and the Tertiary eras? Alright. So you can correlate there if you want, or just remember these dates. Whatever is given, just remember those, especially when it comes to dates, things which you have to just remember. Stick to what is there in NCERT. Don't go outside of that. If you have to leave those questions, let it be. Leave it. But in the desire to be prepared for all those examples, names, scientists kind of questions, you will be wasting time to memorize unwanted stuff. I wouldn't say unwanted, but not so relevant stuff for examination preparation, especially when you have loads of preparation to make. All right. You should know what not to touch and also what to be taken up at what stage. All right. Preparation is strategic. The first mammals were like shrews. Their fossils are small sized. Mammals were viviparous and protected their unborn young inside the mother's body. Mammals were more intelligent in sensing and avoiding danger at least. When reptiles came down, mammals took over this earth. There were in South America mammals resembling horse, hippopotamus, bear, rabbit, etc. Due to continental drift when South America joined North America, these animals were overridden by North American fauna. Due to the same continental drift, pouched mammals of Australia survived because of lack of competition from any other mammal. Lest we forget, some mammals live wholly in water. Whales, dolphins, seals, and sea cows are some examples. Evolution of horse, elephant, dog, etc. are special stories of evolution. You will learn about these in higher classes. Thank you very much. The most successful story is the that is the expression. You will learn about it in higher classes. Okay, good. The most successful story is the evolution of man with language, skills, and self-consciousness. A rough sketch of the evolution of life forms. Their times on a geological scale are indicated in figure 6.9 and 6.10. Origin and evolution of man. About 15 million years ago, primates called Dryopithecus and Ramapithecus were existing. They were hairy and walked like gorillas and chimpanzees. Ramapithecus was more man-like, while Dryopithecus was more ape-like. Few fossils of man-like bones have been discovered in Ethiopia and Tanzania. Important, remember these places, Ethiopia and Tanzania. Fossils of man-like bones. So where do we find fossils? Which were the earliest ancestors of human beings? Between Dryopithecus and Ramapithecus, which is older? Dryopithecus or Ramapithecus? Dryopithecus, because that is more ape-like. Ramapithecus is more human-like. This revealed hominid features leading to the belief that about 3 to 4 million years ago, man-like primates walked in Eastern Africa. They were probably not taller than 4 feet but walked upright. 2 million years ago, Australopithecines probably lived in East African grasslands. Evidence shows they hunted with stone weapons but essentially ate fruit. Then why did they hunt? For fun. Or survival. Killing the animals which tried to kill them, probably. Some of the bones among the bones discovered were different. This creature was called the first human-like being, the hominid, and was called Homo habilis. Homo habilis is the first human-like form that has been recorded. The brain capacities were between 650 to 800 cc. Cc is cubic centimeter. Cubic centimeter is also ml. Cc is equal to ml. Okay. They probably did not eat meat. Fossils discovered in Java in 1891 revealed the next stage, that is Homo erectus, about 1.5 million years ago. You have to write down the sequence of these forms. Which one, what time frame, and what were their characteristics, something to note. Homo habilis. Homo habilis is the tool man. Homo erectus is the upright man. Like that. Homo erectus had a large brain around 900 cc. Homo erectus probably ate meat. The Neanderthal man with a brain size of 1400 cc lived in Near East and Central Asia between 100,000 to 40,000 years back. They used hides to protect their body and buried their dead. Homo sapiens arose in Africa and moved across continents and developed into distinct races. During Ice Age between 75,000 to 10,000 years ago, modern Homo sapiens arose. Prehistoric cave art developed about 18,000 years ago. One such cave paintings by prehistoric humans can be seen at Bhimbekta Rock Shelter in Raisin district of Madhya Pradesh. Important. Question might come. Some historical context to evolution. These things will come. So in this small segment of origin and evolution of man, there are a lot of question possibilities. Just put it out in a table and revise it repeatedly. Agriculture came around 10,000 years back and human settlement started. The rest of what happened is part of human history of growth and decline of civilizations. From prehistoric times, the first settlements, development of agriculture to current human existence, whatever we are today, the study of all that is anthropology. The study of human civilizational development is anthropology. All right. Okay. Done. We have, I think, touched upon everything that needs to be discussed with evolution. So you should be reasonably strong with evolution. We have already discussed many concepts related to chromosomal theory, Mendelian theory, and all that. We will try to quickly summarize most of these concepts. Some gaps we have, there is some things which we have not discussed in detail. We will go into the detail of that. Okay. Chromosomal theory, I have just made some points. Okay, what's okay. These are chromosomes. Okay. Chromosomal theory of inheritance is we can say an extension of Mendelian theory of inheritance in a way, where it supports Mendelian principles, but it contradicts Mendelian principles also. It is deviation from Mendelian principles also. Chromosomal theory versus Mendelian theory, what are the differences? Tell me what both theories agree upon and what they disagree. Genes. Mendel called it factors. We can call it gene or unit of inheritance. Unit of inheritance we'll call it. Unit of inheritance. Mendelian theory call it as factors. Chromosomal theory calls it as genes. A. The recipient should not have capacity to produce antibody against A. That means you cannot give blood to B blood group. So, who can be the recipient? The one who cannot produce antibody A. Who is the one who cannot produce antibody A? The one with A blood group. Who else? AB. This fellow also cannot produce antibody against A. Now, if the donor is B, the recipient should be somebody who will not produce antibody against B. So that will be somebody who has B blood group. He will not produce antibody against B and AB. If the donor is of AB, the recipient should be somebody who should not produce antibodies against both against A or B. Both A and B. So it has to be AB only. If you give A blood group, he can produce antibody against B. If you give B blood group to the recipient, he can produce antibodies against A. So the recipient for AB blood group should be only AB blood group person. If the donor is O blood group, the recipient should be somebody who? This fellow has got no antigen. So recipient can be anybody. If you see, AB can be the donor for everybody. AB can be the donor, AB can be the recipient. AB can be the recipient, O can be the donor. AB is the universal recipient, O is the universal donor. Alright? Be conscious about antigen of the donor and antibody of the recipient. Okay, when you are thinking about a blood transfusion scenario, you should consider the antigen of the donor and the antibody of the recipient. Is that clear? If the recipient has the capacity to produce antibody against the antigen of the donor, that blood should not be given. Alright. So that recipient who does not produce any antibodies is the ideal recipient. You can give any blood. Who is the one who will not produce any antibody? Both of these? AB. So AB can take from everybody. Who is an ideal donor who does not have these antigens? You will not trigger them. The ideal donor is the one without antigens. The ideal recipient is the one without antibodies. We are talking about AB specifically. Correct? So donor, antigen, recipient, antibody. Donor without antigen is the best donor. Recipient without antibody is the best recipient. So O is the best donor, AB is the best recipient. O is therefore called universal donor, AB is therefore called universal recipient. Is this clear? Now, blood grouping. How do we do blood grouping? All this is clear? H antigen significance, I'll tell you. Anybody knows about the significance of H antigen? No? Nobody. Okay. So to check for the blood group of a person, you take a sample of blood. To check for the blood group of the person, what do you want to check in that? You want to check for the presence of antigens. You want to check for the presence of antigens. How do you check for the presence of antigens? You can add antibodies. If you add antibody against a particular antigen, if that antigen is present, it will clump. If that antigen is not present, it will not clump. Clear? So what you will do? You will take, what are the antigens that we want to check for? A, B and D. D antigen is associated with Rh blood group. Whatever we call as positive and negative, presence of D antigen is positive. Absence of D antigen is negative. So there are two components here in blood group, ABO component and Rh component. Unlike the ABO component, absence of antigen will not make the person capable of producing antibody. If A antigen is absent, your body will produce antibody by default. It is ready to produce antibody against A antigen in ABO system. But in Rh, if D antigen is absent, your body does not have the ability to produce antibodies against D by default. You have to expose the blood to D antigen, then it will develop the ability to produce antibody, like regular immunity. Alright. So there is inherent ability to produce antibody against absent antigen of A and B. But it is not the same with D antigen. Alright, that is also significant. I'll tell you why. Is that clear? AB antigen, antigen absent, body can produce antibody by default. You don't have to expose it first. D antigen, Rh group, antigen absent, body cannot produce antibody. Expose the body to antigen, body will develop the ability to produce antibody. Is that clear? Now, to determine blood group, what do? You take 3 slides. These are slides bought in the local market, so they are not standard size. They are of different varying sizes. Cheap slides. Okay, but still glass slides, it will work. Anti-A, you add anti-A antibody in one slide. You add anti-B antibody in the second slide. And in the third one, what do? Anti-D antibody in the third slide. Then here, a drop of blood. On all the 3 slides, you use the blood of the same person. Okay, for every person's blood grouping test, you will take the blood and put blood on all the 3 slides. Then, take one glass, what do you call that? Stick, small glass stick. Mix these two, the blood with the antibody, mix it. Take another glass stick, mix these two. Take another one, fresh one, and mix these two. Observe for clumping. If wherever the clumping happens, it means that particular antigen is present. If clumping happens here, which antigen is present? A antigen is present. If clumping happens here, B antigen is present. If clumping happens here, Rh antigen is present, D antigen is present. So if clumping is seen in all 3, the blood group is AB positive. If clumping happens in the first and third one, The blood group is A positive. If clumping happens in the first and second and not in the third, AB negative. If clumping happens only in the third one, O positive. If clumping does not happen in any of these, O negative. Alright, that's all. Now let us say a person comes with road traffic accident. There is a lot of blood loss. You have to do blood transfusion. You have to do blood transfusion to stabilize the person hemodynamically. You do the bedside blood grouping test. There is anyways blood flowing. You take slides and put it on that and check for the blood group quickly. Alright. There is clumping happening only in the third slide. So as the bedside physician, what do you say? You will instruct the nurse to arrange for which blood group? O positive blood. You will say, sister, arrange for O positive blood. 2 units of O positive blood. There is availability of O positive blood in the blood bank. 2 units of O positive blood comes. You start transfusing the blood. Massive blood clotting reaction happens and the person dies on table. Why? Blood group mismatch. Then you recheck his blood. Same thing happens. No clumping in the first 2 slides. Clumping is seen in the third slide. You check the blood of the donor, what you brought from the blood bank. Same. No reaction in the first 2. The reaction is clumping is seen in the third. Why did the person die? Why did blood clumping reaction happen? Where is mismatch? Where is the mismatch?
Person is RH positive.
Both are RH positive. If both A and B antigens are absent in the blood on the RBC of a person, does that mean there is no antigen on the surface of the ABO system? There is H antigen. Absence of H antigen means that the body is capable of producing antibody against H antigen by default, because that is associated with the same gene I. Now, what if the person who met with the accident, the recipient, did not have H antigen? The donor had H antigen. We did not check for H antigen here. We checked for A and B. In that sense, you got it as O positive. The blood of the donor is O positive. The blood of the recipient is also showing to be O positive. But the donor's blood RBC has H antigen. The recipient's RBC does not have H antigen. There is absence of H antigen. That means the recipient's body has the capacity to produce antibodies against H antigen by default. So you transfuse the blood with H antigen. The immune system identifies it as a foreign body. Massive production of antibody against H antigen, reaction and death. Coagulation reactions and death. This is a special case. This is a special case where H antigen, which is normally present in most of the people's RBC, even in O group people, there will be H antigen. People who have a defect in the I gene, which will not allow them to produce H antigen. If you check them like this, it will show as O positive, O blood group. But H antigen is absent. People whose RBCs, whose gene, the blood group gene, ABO blood group gene, has a defect and will not be able to produce H antigen are called by a special blood group. It is a special blood group and there is a national registry of people with this blood group. If this person dies because of this kind of a reaction, all his blood relatives His kith and kin will be tested for the H antigen to check whether they have H antigen or not. And then if they also belong to that particular group, their name will be put in the registry. What registry is this and what is this special blood group? This special blood group was discovered by an Indian physician. Bombay blood group. This is Bombay blood group. You understand? So there is a registry of people with Bombay blood group. Can you sue the doctor for not checking for H antigen? You can't sue the doctor. Because Bombay blood group is so rare that you will not be in a position to check for H antigen for every person. Alright? H antigen is also tested when there is transfusion required. And if the person turns out to be O positive, O. If the person, the recipient turns out to be of O blood group, as a precautionary measure, when nobody is there to ask whether this person belongs to Bombay blood group or not, H antigen is also tested for. Whether there is H antigen or not. Alright, Bombay blood group, that's all. Clear? And RH group has one, RH group behaves like the regular antigens. That is, if you expose the immune system to this antigen, it will produce antibodies against that. Otherwise, it will not produce. This is important in case of RH negative mother and RH positive father. If the mother is RH negative and the father is RH positive, the baby is likely to be RH positive. The baby can be RH positive or RH negative, but there is a possibility of the baby being RH positive. The baby's blood and the mother's blood are kept separate. Placenta is the barrier. It will allow for exchange of material between the mother's blood and the baby's blood, but there is no mixing of blood happening. Alright? But during the terminal stages of pregnancy, end stages of pregnancy, placental rupture happens. Placenta breaks partially, and that will allow the mixing of blood of the mother and the baby. So the mother's blood can get exposed to the baby's blood. Now if the baby's RBC is RH positive and the mother is RH negative, what will happen in the mother now? The mother's immune system will identify the RH antigen, the D antigen, and will initiate the mechanism to produce antibodies. If you allow this to go on, the mother's immune system will become capable of producing antibodies quickly. It will develop memory of this. And next time there is an exposure, it will rapidly engage the antibody production against the D antigen and push the D antibody into the bloodstream of the baby. Should not happen. So now this is happening in the terminal stages of pregnancy. The first pregnancy, first exposure to the mother. No problem. By the time the mother produces antibodies and pushes it into the baby is delivered, the baby is saved. Subsequent pregnancy, second pregnancy. Now the mother's immune system has a memory of this antigen. The baby's blood gets mixed with the mother's blood. Identification of the antigen. Immediate deployment of the memory cells which are capable of producing antibodies against D antigen. Massive production of D antigen, push the D antibody into the bloodstream of the baby. The baby is delivered, but its blood is loaded with antibody against D antigen. So the baby will go into a clumping reaction. What is this called? Erythroblastosis fetalis. Erythroblastosis fetalis, or the hemolytic disease of the newborn. Those clumped RBCs will be broken down by the baby's system. In the spleen, when these clumped RBCs are trying to squeeze through, they are broken down and all that happens. Lot of blood cell destruction happens. Why erythroblastosis? Erythroblasts are immature RBCs. Because a lot of RBCs are being destroyed, to keep up to the demand of RBC, immature RBCs will come into blood circulation. From the bone marrow, immature RBCs will start coming into the blood circulation. So when you study the blood, when you study the blood, you will find these immature RBCs in the bloodstream, which are erythroblasts. That is why the disease is called erythroblastosis. Erythroblastosis means increase in the number of erythroblasts. Fetalis. Cause is when the baby was a fetus. But the manifestation is when it is already born. Neonate. You could have called it as erythroblastosis neonatalis in the neonate, newborn. But why call it erythroblastosis fetalis? The process began when it was yet to be delivered. All right. How do you protect the subsequent babies from this? How do you prevent this? First baby, okay, taken care of, no problem, it's delivered. How do you protect the subsequent babies? Tell the mother not to go for second baby. We will get attacked. Don't have a second child. What do you do? Anti-D antibodies. As soon as the baby is born, the mother is injected with anti-D antibodies. It is like passive immunization, anti-D antibodies. So what will these antibodies do? They will go and find D antigen wherever in circulation and bind to them and remove them from circulation. Before the mother's immune system can identify D antigen and produce antibodies against it and remember, this will sneak it out. This will sneak it away. Got it? It's like, have you seen the movie RRR? Recently you watched it here or before only you had watched at home? RRR. This fellow comes to Delhi to save the girl. One of his accomplices gets noticed by the policeman, and before the policeman could catch him, he sneaked away. Same. Before the body can start producing antibodies, antibodies against antigen, you remove the antigen from the body so that the body cannot produce the antibody. That's all, okay? So this is with blood groups. Enough. All right? So deviations again. Deviation from Mendelian principles. We started with Tom and Jerry and we are coming here. Blood group we elaborated. One more aspect of genes, chromosomal theory we need to understand is gene interactions. There are supplementary genes, there are complementary genes, epistatic genes, lethal genes. What are these? What are supplementary genes and complementary genes? See, now so far we have seen the variations in individual genes. Dominant expression, recessive expression, codominance, incomplete dominance, all of these are associated with one gene. What if genes interact? Do the genes interact? Mendel does not talk about gene interaction. In Mendelian principle, there is no concept of gene interaction. In chromosomal theory, there is concept of gene interaction. Gene interaction. See, it's like you have one function of your own, that is when you are dominant, another function when you are recessive. Both the genes have individual functions of their own, but when they come together or when in the presence of another gene, there will be a new expression that is produced. All right? Gene interaction, that is supplementary gene. When the two genes are supplementary to each other, when the genes have individual expressions when they are separate, but when they interact, there is a new character that is produced. Comb on chicken. There is P comb controlled by gene P. There is a rose comb controlled by gene R. When gene P is recessive and only gene R is active, you produce rose comb or you don't produce rose comb. Both of, I'll just show you. You know what is comb in a chicken? That what you have here on the head, that red color thing. Okay. There is one gene R, there is one gene, gene P. Dominant character and recessive character. For gene R, the dominant character is rose comb. For recessive character is straight comb. For gene P, the dominant character is P comb. The recessive character is straight comb. Both the genes are present. Both the genes are present in the chicken. Now, if the combination is what comb will you see? This fellow, gene P, is trying to produce straight comb, but this fellow is producing rose comb. So what comb will be there in the chicken? It will be the rose comb. When you have a dominant allele for gene R and recessive alleles for gene P, you will have rose comb. It can be this also, capital R, small p, small p. Even in this case, it will be rose comb. When both are recessive, small r, small p, it will be straight comb. Both of them are producing the same character. Correct? Reverse it. When it is small r, capital P, or small r, capital P, small p, in both these cases it will be P comb. Small r, small p, straight comb. Now the supplementary nature of these two genes will come into picture when both of them have at least one dominant allele. That means when both the genes have at least one dominant allele, a totally new character will appear, phenotype. It is called walnut comb, walnut shaped comb. So walnut comb is an expression of gene interaction. And what is the kind of interaction? Supplementary interaction. Got it? Supplementary interaction. A supplementary task is an additional task. Correct? When you write supplementary examination, what does that mean? You have already written that examination earlier. In addition to the one that you wrote in which you did not pass, you are writing one more. Supplementary. Got it? In addition to something. So individually the genes have their own characteristic that they will control, but when they are together, there is a new character that comes up. These are supplementary genes. These are supplementary genes. Understand? So the example I would like to take for this is, you beat up one teacher when you are independent. Your friend beats up one teacher when he is active. When both of you are together, you beat up a different teacher separately. So you don't like, or it's like, why beat up unnecessarily? Okay. Let's say you don't like mathematics teacher, your friend doesn't like chemistry teacher, but when you are together, you don't like biology teacher. This is supplementary genes. Complementary genes is, when you are separate, you are not expressing dislike on anybody. When your friend is separate, your friend is not expressing dislike on anybody. But when both of you are together, you are expressing dislike on somebody. So the expression of a character requires the presence of both genes. That is complementary. That is complementary genes. You understand? Here, supplementary genes will have their own independent expression, but when they are together, when I say together means what? Both of them have at least one dominant allele. Both of them having at least one dominant allele is being together. Either of them or both of them not having any dominant alleles is being separate. That's all. Understood? This is supplementary genes. Complementary genes is, when they are separate, there is no expression. When they come together, there is some expression, a new expression. That is complementary gene. What is epistatic gene? Epistatic gene.
It must
Masking effect. They are masking genes. They are masking genes. All right. Gene 1 has its own expression. Let us say it is expressing something. We will relate it to writing. Let us take away the negative references. Supplementary gene is when you are alone, you write a story of your choice. When your friend is alone, he writes a story of his choice. When both of you are together, you will write poetry. This is what gene? Both of you are supplementary genes. When you are alone, you don't write. When your friend is alone, you don't write. When both of you come together, you start writing. This is complementary gene. Complementary gene. You are normally writing. You are writing prose or poetry based on your mood. That is based on your genotype. Dominant or recessive. In the presence of your friend, you will stop writing. This is dominant epistasis. You will be writing, normally you will be writing, but in the absence of your friend, you will stop writing. When your friend is there, you will write. Alright? You are writing normally. For you to write, if the presence of your friend is required, and in the absence of your friend, you will stop writing. No, presence doesn't matter. If your friend is absent, you will stop writing. Then the effect of stopping your work by your friend is recessive. Absence is recessive. Presence is dominant. So in the presence of your friend, if you stop writing, you are experiencing dominant epistasis. Epistasis. If you are experiencing epistasis or suppression of your work in the absence of your friend, then it is recessive epistasis. Recessive epistasis. All right. If the presence of your friend kills you, your friend is a lethal gene. Okay. If the absence of your friend kills you, your friend is still a lethal gene, the lethality being in the recessive state. If the presence of your friend kills you, then lethal gene in a dominant state. When your friend is dominant, when the gene is dominant, it will kill. When the gene is recessive, if it kills, it is a recessively lethal gene. It kills when it is recessive. Understand? Clear? Earlier questions used to come on the phenotypic ratio of gene interactions. Phenotypic ratio of gene interactions. If it is a supplementary gene, what will be the phenotypic ratio? If it is a complementary gene, what will be the phenotypic ratio? What will be the phenotypic ratio if it is, let's say, dominant epistasis? What will be the phenotypic ratio if it is recessive epistasis? Let us work it out. Let us see. What is the phenotypic ratio and when two genes are involved, when we talk about it in Mendelian terms, what kind of cross that would be? If you are working out the genotype and phenotype of two genes being in question, what type of cross that is? It is a dihybrid cross. Now here also in gene interaction, we are considering two genes. So how do we represent the two genes? We represent it as dihybrid cross only. We represent it as dihybrid cross. All right. So we will work out. You want to work out? I will give you the genes. You work out and tell me the, or does anybody remember the phenotypic ratio of supplementary genes? What?
9:3:4 for supplementary and for complementary.
9:3:4.
And for complementary 9:7.
Complementary 9:7. Okay. Epistasis? All right. Okay. Have I told you everything that I wanted to tell about Tom and Jerry? Incomplete dominance. Incomplete dominance phenotypic ratio. Monohybrid. 1:2:1. Codominance. 1:2:1. Blood group is also an example of multiple allelism. Not only codominance, it is also an example of multiple allelism. Height and skin color are examples of polygenic inheritance. Not only polygenic inheritance, they are also examples of quantitative inheritance. Quantitative inheritance. Again, Mendelian principle is qualitative. Mendelian inheritance is qualitative, but these are quantitative inheritance. So which one will give you graded expression? Qualitative inheritance or quantitative inheritance? A spectrum of phenotypic expression is possible in qualitative or quantitative? Quantitative. If it is qualitative, it is just presence or absence. Presence of dominant allele, full expression. Absence of dominant allele, no expression. A range of expression is possible when the number of dominant alleles will determine the extent of expression, which we see in height. Epistasis. What is pleiotropy? We discussed that. A single gene affecting multiple traits. Epistasis, masking genes. Epistatic genes are masking genes. The genes which will suppress are called epistatic genes. What do you call the gene that gets suppressed? The one that gets suppressed is hypostatic gene. Epistatic gene is the one that will suppress. Hypostatic gene is the one that will gets suppressed. In most of the television dramas, you see epistasis in action. Sometimes the epistatic gene can turn out to be lethal gene also. How? Can you give example? Sas Bahu dramas. Sas Bahu dramas, it is epistasis. Okay. Anyways, epistasis only, no? Somebody is trying to dominate. Dominant epistasis, 12:3:1 phenotypic ratio. Recessive epistasis, 9:3:4. You can work it out. All right. Is inheritance of characters always chromosomal? Is there cytoplasmic inheritance noted? There is something in your body which is exclusively maternal in origin, no paternal contribution, irrespective of whether it is male or female. Exclusive maternal origin is mitochondria. Mitochondria has its own genetic composition also. The mitochondria that you find in the ovum is, the mitochondria that you find in the zygote is of the ovum. It's of the ovum. So if you see that, the entire energy machinery in your body is maternal. The power is coming from the mother, not just the life force. So you can consider it as a life force, creative force. Mother gives you the mitochondria. The mitochondria of the sperm is where? Where is the mitochondria of the sperm? Where is the mitochondria of the sperm? It's in the middle piece as the jacket. Mitochondrial jacket the sperm is wearing, the Nebenkan, or the machete, the mitochondrial coil, that will help in propelling the tail of the sperm for the sperm to move. All right. Lethal alleles, gene interaction, supplementary genes 9:3:4, complementary genes 9:7. Okay. Yeah, I think we have done most of it. Any specific doubts, please ask. You can work out with random examples. Let's say, supplementary genes. Let us say there is gene A, gene A and gene B, gene A and gene B. There is phenotype A or whatever. In supplementary genes is when both of them are present, there is a different expression. Correct, no? When both of them are present, there is a different expression. So you have gene A and gene B, the first generation is all homozygous dominant. So what we go for second generation, the genotype of the two that you will cross will be this one, no? This will be the genotype. This will be the genotype. So we have to look for those phenotypes which has expression of A, those which has expression of B, and those which has a new expression. That's all. Correct, no? We will not write the phenotypes. AB, 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 and AB. These are the four gametes. Same here. AB, 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 AB. We will directly write the phenotypes. Okay, we want phenotypic ratio, no? We want phenotypic ratio. So, looking at it, we will just mention what kind of character will be expressed. Will it be character A or character B, or the supplementary character we will call it as character X. Here, what will be the character that will be produced? Both are dominant, both are present. So character X. If you have at least one dominant allele for both, you will get a new character X. Here, A or B or X? X. Here, X again. Here, X. Correct? Here, X. Same, this also. So this one, A, good. This one, X. Here, A. Here, X. Here, B. Here, B. Here, A. Here, B. Here, both are absent. Huh? What will be produced? When both are absent, what will be produced? Whatever is the recessive character. Whatever is the recessive character. Alright? Let us say, we will mention it as AB probably. The recessive character, whatever is the recessive character that is produced. for when both are absent. So count number of eggs: 1, 2, 3, 4, 5, 6, 7, 8, 9. Count the number of A's: 3. Number of B's: 3. Number of recessive: 1. What is the supplementary gene ratio? 9:3:3:1.
9:3:4.
9:3:4. We will do it with pea comb and rose comb, the same example that we took. All right. Okay, let us consider the supplementary character to be walnut. 9:3:4. The premise of cross is wrong. All right. It is not this. Then this will be walnut only, no? Both of them would be walnut comb. We are crossing both walnut comb. Okay. One is rose comb. We will do it this way. One is rose comb. One is pea comb. All right. You have a rose comb chicken crossed with a pea comb chicken. You have a rose combed chicken crossed with a pea comb chicken. Then you work it out. You will get it. Then if you work it out, you will get it. 9:3:4. Something missing, no? We will not do this now. Okay? Let me revisit the premise of calculating this supplementary gene and complementary gene. Then we will do the calculation the next class. All right. Yeah. Done. What next? What topic do we discuss next?
Muscle contraction. Muscle contraction.
Muscle contraction. Okay. We will read out from the book first. 10 minutes we have. We will try to read out without any explanation. Understand whatever is given in the book, and in the next class we will study that. Muscle structure we have already studied. We have left out muscle contraction. Give me the 11th standard textbook. We will just read that portion of the write upon muscle contraction. Do you remember the structure of the muscle? Cross section? Page number 221. Structure of contractile proteins which will extend to muscle contraction mechanism also. Just 2 pages. We will read it. I will read it slowly. So try to pay attention. Then we will expand it as the first thing in the next class tomorrow. Structure of contractile proteins. Each actin or thin filament is made of two filamentous actins helically wound to each other. Each F-actin or filamentous actin is a polymer of monomeric globular actins. It is like beads of, it's like a chain of beads. Okay, each bead in the chain is a globular actin and the entire chain is the filamentous actin. Two filaments of another protein, tropomyosin, also runs close to the F-actins throughout its length. It's like a binding thread on these two filaments. In the resting state, a subunit of troponin masks the active binding sites for myosin on the actin filament. So the complete unit of contraction in the thin filament is the string of beads, which is the F-actin, filamentous actin. Each bead is the globular actin. They are wound to each other, tied around by tropomyosin. Troponin is blocking the site of attachment of the head of the thick filament or the myosin filament. So if you want attachment of the thick filament to the thin filament, you need to move that troponin so that the active site gets exposed. We will see how that happens. Each myosin or thick filament is also a polymerized protein. Many monomeric proteins called meromyosins constitute one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail. The former being called the heavy meromyosin, that is the globular head is the heavy meromyosin, and the latter or the tail is the light meromyosin. The heavy meromyosin component, that is the head and short arm, projects outwards at regular distances and angle from each other from the surface of a polymerized myosin filament and is known as cross arm. It is like a folded arm, a chain with some folded structures there. All right. You make a rope of this meromyosin, which is like a 120-degree angled rope, 120-degree angled structure. You join all of them at the neck and bind them to the tail. All right, one by one. So throughout the entire length, you will have these extended heads from the rope. All right. The globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. So it is an enzyme, it has an enzymatic action because it's a protein, it functions like an ATPase enzyme. So the head attaches to the thin filament on the active site, binding site. ATP is broken down, energy is used for flexion of the neck. Which pulls the thin filament, which is attached to the Z line, pulls the sarcomere closer to each other, shortening the length of the sarcomere, thereby bringing about contraction. Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of a muscle fiber takes place by the sliding of the thin filaments over the thick filaments. Muscle contraction is initiated by a signal sent by the central nervous system via a motor neuron. A motor neuron along with the muscle fibers connected to it constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle, sarcolemma is the cytoplasmic membrane, is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter, which is acetylcholine, which generates an action potential in the sarcolemma. Now we know what an action potential is, change in the voltage of the membrane there. This spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. Increase in calcium ions level leads to the binding of calcium with a subunit of troponin on actin filaments and thereby removing the masking of active sites for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed active sites on actin to form a cross bridge. This pulls the attached actin filaments towards the center of A band. The Z lines attached to these actins are also pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. It is clear from the above steps that during shortening of the muscle, there is contraction, the I bands get reduced, whereas the A bands retain the length. The myosin releasing the ADP and inorganic phosphate goes back to its relaxed state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back into the sarcoplasmic cisternae, resulting in the masking of actin filaments. This causes the return of Z lines back to their original position, that is relaxation. The reaction time of the fibers can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscle contains a red colored oxygen storing pigment called myoglobin. Myoglobin content is high in some of the muscles which give a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscles. On the other hand, some of the muscles possess very less quantity of myoglobin and therefore appear pale or whitish. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. The last paragraph mixes up a lot of things there. Talks about myoglobin, sarcoplasma, the calcium, red fiber, white fiber and all that. Okay. Now, the simple mechanism is what I told you in the beginning. There is actin filament, there is myosin filament, thin filament, thick filament. Thick filament can attach to the thin filament and pull the thin filament by the flexion of its head. To do that, it needs energy. Energy comes from the binding of ATP to the head itself, which is an enzyme, ATPase enzyme. Breakdown of ATP provides energy. Flexion of the neck pulls the thin filament. But the thin filament to be pulled requires attachment of the head of the thick filament to the actin thin filament. What is preventing the attachment? Troponin is preventing the attachment. You need to remove the troponin. Who will remove the troponin? Calcium. Where is the calcium coming from? Sarcoplasmic reticulum. Sarcoplasmic reticulum. All right. Now, we experience, so when ATP again goes and binds. When there is ATP hydrolysis, it gives energy for the actin thick filament head to go and attach to the troponin, attach to the binding site. When ATP comes and binds to the head and it does not get hydrolyzed, then the head is detached from the binding site. All right. Calcium has to be removed from this sarcomere and pushed back into the sarcoplasmic reticulum. So when calcium is pushed back, pumped back into the sarcoplasmic reticulum, and when ATP goes back and goes and attaches to the head of the thick filament, not allowing further, undergoing further hydrolysis, the relaxation happens. But as long as there is trigger for contraction, where is it coming? Neuromuscular junction, release of acetylcholine. Acetylcholine binds to the receptor on the muscle sarcoplasmic membrane. That will stimulate the internal second messenger system, cyclic AMP dependent second messenger system, to go and activate the release of calcium from the sarcoplasmic reticulum. Calcium is released. That goes and binds to the troponin, exposing the active site. Parallelly, the message is given to the head of the thick filament to hydrolyze ATP. ATP is hydrolyzed. The thick filament uses the energy to attach to the active site, pull the thin filament. Sarcomere is shortened. Sarcomere is shortened, bringing about contraction. Now the stimulation by the neuron at the neuromuscular junction is removed by acetylcholine getting cleared off. What will clear off acetylcholine from the neuromuscular junction? There is an enzyme, acetylcholine esterase. Acetylcholine esterase will act on the acetylcholine in the neuromuscular junction and clears it off. So when there is no stimulation of the receptors in the neuromuscular junction, the second messenger system gets deactivated. When the sarcoplasmic reticulum is not being stimulated to release calcium, the counter mechanism gets activated, that is clearing calcium from the sarcomere back into the sarcoplasmic reticulum. Calcium gets back into the sarcoplasmic reticulum. troponin comes back and binds to the active site, causing relaxation. All right. What is the disease where there is problem of acetylcholinesterase? Acetylcholinesterase is defective. We have discussed this. What is the disease? Acetylcholinesterase is absent or becomes less, so it will lead to constant involuntary contractions of the muscles. What disease? Come on, check out from your nose. Huh? Not tetany. Tetany is because of calcium deficiency. Myasthenia gravis. Myasthenia gravis is due to defect in the acetylcholinesterase, either because of there is no stimulus for that to continue contraction. Why? Detachment of calcium from the actin thin filament from the active site of the thin filament requires an adequate level of calcium in the blood. If there is shortage of calcium, the mechanism is that the calcium goes and remains bound to the actin site, bound to the active site of thin filament. So contrary to logic that we feel that the calcium deficiency should remove calcium from the binding site, clearance of the calcium from the binding site and pushing it back into the sarcoplasmic reticulum requires calcium in adequate levels in the bloodstream. All right? If calcium deficiency is there, this process gets hindered. Understand? Okay. Right. What topic for tomorrow? What? Codons, genetic code. Okay, that will be a small topic. Then, anything else? Dihybrid cross. Okay. Done. And we will do these calculations also. I'll just rework on this complementary and supplementary gene ratios, and we will do that with an example. Countercurrent mechanism. Nephron. Countercurrent mechanism also is one complex topic. We will do that also. So we have 3 topics. Countercurrent mechanism, dihybrid cross of Morgan, and genetic code. Done.